وفي ايه في النت يا رب؟ Okay, can you all hear me?
Yes.
Okay. Give me a second, I'll share my screen. Okay, can you see my screen now? Hello?
Yes, we can.
Okay, give me a second because I need to. Okay, what will we take today? We will start a new lesson. This lesson is really short and it is really easy. It is called biological molecules. It doesn't want to open, just a second. Okay, biological molecules mainly talks about your food. It mainly talks about your food. Okay, don't worry about recording the session. I'll be recording it for you. The biological molecules, that lesson, it talks about the molecules in your food, the carbohydrates, the proteins, etc., etc. Where is it? Movement in and out of cells. We will start the biological molecules is lesson 4. It's only 5 pages. Okay, and again, most of the questions in this lesson, they are found in paper 6. Okay, so biological molecules. It's a very, very interesting lesson, and you will notice that we already explained a very important objective before. In biological molecules, you have to study the food tests, and we already discussed the food tests, and I'm going to explain it again. So the biological molecules, what does it talk about? It talks about the food in your, the molecules in your food, the carbohydrates and the proteins and the lipids. Now, before you ask me what are lipids, who knows what are lipids? So we will be studying the molecules found in your carbohydrates. In case you're not sure what are carbohydrates, carbohydrates are the sugars. Okay, we have complex sugars and we have simple sugars. And the proteins, of course, you know what are proteins, fine. And the fats or the lipids. Now, these biological molecules that we are talking about, the carbohydrates and the proteins and the lipids, they are all called organic because they contain the element carbon. Okay, so you need to know in biology or chemistry, by the way, this is organic chemistry, Any molecule containing the element carbon is described as organic. Any molecule that has no... Okay, I think I have to mute you all.
Hey guys, good morning. Or just evening, but
Okay, I muted you. If you have a question, raise your hand and I will unmute you. Good. Now, again, going back, we were talking about the biological molecules. We were talking about the carbohydrates and proteins and the fats, and they are described as being organic. Why organic? Because they contain the element carbon. If they do not contain carbon, then they are considered inorganic. Now, if you want to answer the question, raise your hand and I will unmute you. Can someone tell me what is the chemical formula for water? Water is what? Raise your hand if you want to answer. Where is Habiba? Oh my God. Habiba, okay. Now, Habiba, you can unmute yourself and tell me what is the molecular formula for water?
H2O.
H2O, very good. Is there any carbon
H2O.
involved? Yes, you are correct. Is there any carbon involved in the molecular formula of water? So you're telling me that H2O is the molecular formula for
No.
water, and you're very correct. Good. So no
No.
carbon. As long as there is no carbon, this water is considered as organic or inorganic?
Inorganic.
Inorganic, okay? So these terms are important. We are talking about proteins, carbohydrates, carbs for short, and lipids or fats. Those three are the major biological molecules found in your body and in your food, and they all contain carbon. So they are all described as being organic. Now, any molecule that has no carbon, such as water, it has no water, then it is considered inorganic, even though water is very important, but it is described as being inorganic because it has no carbon. Now, let's talk about each one in detail, the proteins and the carbs and the lipids. Let's talk about the carbohydrates because these are really important. Carbohydrates, who wants to raise their hands and tell me what are the elements found in carbohydrates? If you want to raise your hand and tell me, let me see. Who has their hand raised? Layla. Layla, can you tell me, my dear, what are the elements found in carbohydrates?
Carbon.
Very good.
Hydrogen.
Carbon. Yes.
Oxygen.
And what did you say?
Oxygen.
Excellent. So the elements found in carbohydrates are the carbon and the hydrogen and the oxygen. And can you tell me what is found in lipids? Layla also, can you tell me what are the elements found in lipids?
It's the same thing.
Excellent. Again, lipids contain carbon and hydrogen and oxygen. Now, Layla, can you tell me what are the elements found in proteins?
The same thing, but with nitrogen.
Perfect. Proteins also contain carbon and hydrogen and oxygen in addition to nitrogen. So all three are considered organic molecules or not?
Yes.
Yes, they are all considered organic molecules and they all have the same elements, but of course they have different molecular structures. That is why we have three different biological molecules, carbohydrates and lipids and proteins. And the protein is special or easy to identify because proteins contain nitrogen. So you are not requested to know the molecular formula for now because you're still in O levels. But you will have to know it later. This is not important. For now, you need to know that the proteins, they have nitrogen, and this is the extra element that is found in proteins and not found in carbohydrates and in lipids. Very good. Now, carbohydrates, who can tell me what are the different types of carbohydrates? Who wants to raise their hand? Lily, can you unmute yourself or I have to unmute you? Let me see. Where is Lily? Yes, Lily, my dear, can you tell me the types of carbohydrates? Lily? Hello? Who else wants to answer? What are the different types of carbohydrates? Who knows? Dalia, where is Dalia? Dalia is here. Yes, where is Dalia? Yeah, Dalia, can you tell me what are the different types of carbohydrates?
Yes, monosaccharides, disaccharides, and polysaccharides.
Okay, that's excellent. That's very advanced. But before we move into that, carbohydrates are divided into the simple sugars, yes? And simple sugars and what else? Complex.
Reducing sugars.
Very good.
Complex.
The complex sugar.
Okay.
Yes, okay. So we have, and it's important to understand that because they have very different molecular structures, they have very different functions, and they have even different food tests. Now, the simple sugars, I want you to always be familiar with the term reducing sugar. Okay, so simple sugars are also called in all examination boards as reducing sugar because a lot of you read the term reducing sugar and you don't know that they are referring to simple sugars. For example, glucose. The example that you all have to know about. Glucose is a reducing sugar. Glucose is a simple sugar, okay? And a glucose molecule, just like Dalia said, a glucose is also described as something called monosaccharide. Now, be patient and you will discover that these terms, they sound complicated, but they're very easy. Mono means what? What's the number mono? Mono is number
One.
what? One. Very good. What is saccharide? Saccharide means what in English? Who knows?
Sugar.
Yes, very good. Saccharide means sugar. So monosaccharide means how many sugar units?
One.
One. Okay, so it's really easy. So a glucose is a reducing sugar or a simple sugar, and it is also described as a monosaccharide. It is made up of one molecule, one sugar molecule. Okay, very good. Now, sometimes carbohydrates can be disaccharides. Can you guess what is a disaccharide?
Many
Very
sugars.
good. Two sugar molecules, two sugar units. And the only example that you need to know for your syllabus in a
Sucrose and maltose.
Fructose.
very good disaccharide. Disaccharide means it's a sugar made up of two sugar units. Example, sucrose. And we will study more about sucrose when we study plants. Very good. Now going back to glucose, because glucose is very important, or the reducing sugar. If you want to know why is it called a reducing sugar, because who knows what is a reduction reaction in chemistry?
Something that takes away the oxygen from a reaction.
Very good. And it donates electrons. So the reducing sugar, this glucose, which is a reducing sugar, it releases electrons or it donates electrons. Well, this is too advanced for you. This is organic chemistry. We don't have to know that. Glucose is the reducing sugar and it has a specific food test. What is the food test for glucose?
Benedict.
Very good. The Benedict test. In case you forgot, the Benedict test or the Benedict solution, it is a ready-made solution in the lab. We will not prepare it. We don't have to know its composition. If we have an unknown solution, this is an unknown solution, and I want to know if it contains glucose, what do I add?
Few drops of Benedict test.
Perfect. I will add a few drops of Benedict test. And what is the starting color of the Benedict?
Blue.
Blue. Okay, so I'll color it in blue. And then this is the only food test that has to be what? Who remembers?
Heated.
Excellent. It has to be heated in a water bath of a temperature at least from 70 to 80 degrees Celsius. If this unknown sugar contains glucose, then the color change will be from blue to
Brick red.
Brick red. Very good. And the color goes through several changes, like it changes to green first and then yellow and then brick red. And of course, the darker the color, the more it contains glucose sugar. Now, let's shift to paper 6 or practical questions. When the color is the result of your experiment, this result, is it qualitative or quantitative?
Qualitative.
Very good. Qualitative. Qualitative. Coming from the word quality. Okay, I'm going to look at color, which is accurate or inaccurate?
Accurate.
Inaccurate. Now, how can I improve or increase the accuracy of my results?
Colorimeter.
Using the
Excellent.
colorimeter.
You are amazing. In case you forgot also, we use a machine called a colorimeter. I think I showed you the picture in the lab, in class. Okay, and it will change the color into a number. So no one will argue whether this color is light or dark. Very, very good. You are amazing. Okay, let's go back to our lesson so that we don't jump. Okay, we are talking about carbohydrates. Carbohydrates are made up of simple sugars. Glucose is a simple sugar, a monosaccharide. When two glucose molecules join together, maltose is formed, and this is an example of a disaccharide. So I want you to know 2 examples for disaccharides. Sucrose is an example, okay? And maltose is another example. Just names. You don't have to know any molecular structure or anything, okay? When lots of glucose molecules join together, we will make complex carbohydrates. Now let's shift to complex carbohydrates. Now we were talking about simple carbohydrates and we said 1 sugar unit, example, glucose is a monosaccharide. 2 sugar units, example, maltose and sucrose, and 2 sugar units are called disaccharide. Okay, let's say we have much more than 2. We have thousands of glucose units. Glucose. Exactly. Many glucose, many, okay? It's going to be called a polysaccharide. Again, the term seems complicated, but it's actually very easy. Poly means many, and saccharide means sugar. So I have many sugar molecules, I'll make a polysaccharide. Now, what are the examples of polysaccharides that you know about?
Cell wall.
My dear, cell
Cellulose.
is made up of a polysaccharide. Yes, what is it?
Cellulose.
Excellent. Cellulose. Cellulose is a polysaccharide that is found in plants. Where exactly in plants? You just said it.
Cell wall.
The cell wall. Perfect. And is it freely
makes up the-
permeable?
Really?
Yes, you are correct. Is it freely? It's freely permeable. Perfect. And is it strong or weak?
Strong.
Strong and inelastic. Inelastic, it does not stretch. It provides support and protection against turgid pressure. I think this is all clear for everyone. If everyone has any questions, ask me. Now, another Yes, my dear. Of course. Now, do you understand that many glucose molecules joined together forms a polysaccharide?
Yes.
Okay, good. Polysaccharides, we will study 3 examples. Example number 1, my dear, is the cellulose. The cellulose is a polysaccharide. In other words, the cellulose is made up of many sugar molecules bonded together. Okay? Any questions up to now? Good. Now we want to know, where is this cellulose found? It is found in the plant cell. Remember the plant cell? This is the cellulose cell wall. This cellulose is freely permeable. In other words, anything can move in and out of the cell wall. Its function is not to control what moves in and out of cells. Its function is to provide support and shape to the plant cell and protection. What does it protect it from? Who remembers?
Bursting.
Very good. Excellent. It protects it from the high pressure that builds up inside the vacuole, which is called
Turgid or hydrostatic pressure.
Okay, you are more than amazed. More than amazed. Excellent. Now we are, my dear, who asked, is any questions about cellulose? Is cellulose clear now?
Oh, darn.
Okay. Now another polysaccharide that we will also study that we are very familiar with is starch. Now, starch is a polysaccharide. It is a complex sugar. It is also made up of many glucose molecules bonded together. Starch is found in your food. It is found in food like rice, like bread, like pasta, macaroni, okay? And starch is how plants store carbohydrates. In other words, this is the plant cell. I don't want any confusion. This is the plant cell. This is the cell wall. The cell wall is made up of what? Which polysaccharide?
Cellulose.
Excellent, cellulose. Okay, and then we have the plant cell with all the organelles, and the plants store starch. So these are starch granules, okay? So the plants, they store the starch. So I don't want any confusion. The cell wall is made up of cellulose, and where is the starch found? Inside the plant cell. It stores carbohydrates in the form of starch, okay? If we want to clarify matters, plants, of course, they do photosynthesis, carbon dioxide and water to give us what?
Chloroplasts.
Chloroplast and glucose.
This happens in the chloroplast, yes, in the presence of light.
Oxygen.
Yes, it gives us glucose and what? Oxygen. Do you all agree? Anyone has any problems with this equation? Anyone has any questions?
Oxygen.
Yes, and oxygen. Now, the glucose is the product of photosynthesis. After the plant produces the glucose will be stored. But the plant will change the glucose to what in order to store it?
Starch.
To starch. Well done. Because some students, I'll repeat it, my dear, if you're confused. Some students, they get confused between starch and cellulose. Both of them are found in plants. But the cellulose, where is it found? Where in the plant?
The cell wall.
The cell wall.
Outside the cell.
Exactly. And the starch, this is how the plants store carbohydrates. They store it as starch. Okay? So the starch can be described or defined in biology as storage carbohydrate in plants, not in animals. We don't store carbohydrates in the form of starch. We eat it and we digest it into glucose. But we do not store our carbohydrates as starch. We store our carbohydrates as what?
Glycogen.
Okay, perfect. Some of you know it, some of you don't. No problem. The third complex molecule I will discuss today, or the third polysaccharide, is glycogen. Glycogen is simply, yes, my dear. Anyone has
Didn't
questions?
understand the previous point. You have this.
Okay, I'll repeat. Fine. Photosynthesis. Do you know what is photosynthesis? Do you know what is photosynthesis, or you have any problems with photosynthesis? Photosynthesis is how plants make their own food.
We do.
Okay? Photosynthesis is when the plant absorbs carbon dioxide from the air and water from the soil in the presence of light, and this happens also in the chloroplast, to give you products. These products are glucose and oxygen. Who is asking? Whoever is asking, do you have any problems with photosynthesis with this equation?
Me.
Okay, do you know what is photosynthesis and you understand it now?
Yes.
Okay.
The problem is not here.
The problem is what?
The problem is how do the plants store glucose as starch?
Yes, I will tell you. Glucose is the product of photosynthesis. Glucose is converted to starch. After it is converted to starch, it will be stored. How it is converted to starch is not your problem. You don't have to know how it is converted to starch. It will actually change from a monosaccharide to a polysaccharide. Many glucose molecules will bond together, glucose, changing into starch. And then when we change it into starch, we will store it. Better now?
Yes.
Good. Thank you so much.
The point is the carbohydrates.
What's wrong with the carbohydrates?
How are they stored? يعني,
In plants as starch.
so carbohydrates also, they are stored as starch.
Yes, we are talking about carbohydrates. We're not talking about anything else.
Okay.
Okay, and this is how carbohydrates are stored in plants. In our bodies, carbohydrates are stored in a different manner. We are who's asking?
Mimi, I was the
Okay, my dear, tell me, are you a plant?
No,
You're not a plant. So do you do photosynthesis?
no.
No. How do we produce energy? What is the process that happens in our bodies to generate energy?
So we are consumers. We take food from the outside and
Perfect.
we convert it to energy and
Yes, stop here. How do we convert the food to energy? How do we break down the food to release energy? We took it from the very first day of class. Characteristics of living organisms. All living organisms, they do what in order to produce energy? Help him. Who will help him?
respiration.
Respiration, my dear. Respiration, okay? We are consumers, just like you said. So I'm talking about animals, okay? I'm talking about us. We are animals. Consumers, they eat food. This food is combined with oxygen, and it is broken down to give us carbon dioxide and water and energy. If we eat starch, we eat rice, yes, and we eat bread. Our body will break it down, break it down, break it down, break it down until it turns into glucose. Yes?
Yes.
We will use some of this glucose in respiration. And some of it will be in excess because sometimes you eat lots of food. You don't use it. This glucose, the excess, will be stored. Do you all agree?
Yes.
Good. How is it stored in your body? Is it stored as starch?
No.
It is not stored as starch. It is stored as another carbohydrate called glycogen. So we store carbohydrates in a different way. Plants store it as starch, and animals and fungi store carbohydrates as what?
Glycogen.
Okay, I hope it's clear. It's really easy. Let's summarize carbohydrates in case anyone is still confused. We are talking about the organic molecule carbohydrates. Carbohydrates contain carbon and hydrogen and oxygen only, the three elements in carbohydrates. Carbohydrates are either simple or what? Complex. Simple example, the monosaccharide and the, wake up with me, and the what?
Disaccharide.
Give me an example for monosaccharide.
Glucose.
And glucose is also described as reducing sugar. Give me two examples for a disaccharide.
Sucrose and maltose.
Okay, sucrose and maltose. Those that are all asking about notes, you can copy them as we're going on. Complex carbohydrates. Give me three examples.
Starch, cellulose,
Very good.
glycogen.
Excellent, you're amazing. Starch and glycogen and cellulose. Which two complex carbohydrates? Which two complex carbohydrates are found in plants?
Starch and cellulose.
Lovely. Which is found in our bodies and in the body of fungi?
Glycogen.
Glycogen. Okay, are carbohydrates clear? Any questions? Okay.
No, they're clear.
We're not done with carbohydrates. We will be done when we discuss the food tests. So there are two types of food tests for carbohydrates. There is a food test for simple or reducing sugar. Simple or reducing sugar. You will remind me because I already explained it in class. Reducing sugar, such as the glucose. What is the test for the simple or the reducing sugar?
Benedict
test. And there's another test for starch.
Iodine.
Iodine, very good. I will apply drops of iodine on my potato and the color will change. What is the color of the iodine?
Brown.
Very good. Yellow, brown. If starch is present, it will change to what?
Blue.
Blue,
Blue
black.
black.
Blue, black. Very good. And I'll send you the video I showed you in class. So I think we're done with starch completely. Let's look at the notes so we don't miss anything. Monosaccharide, single sugar, disaccharide, 2 sugar units. Example, sucrose and maltose. Mono and disaccharides are soluble and they taste sweet. Okay. Maybe we missed this sentence. Think of the sugar that you add to your tea. It is very soluble in water and it tastes sweet. Polysaccharides, many sugar molecules joined together. They are not soluble in water and they don't taste sweet, just like rice. Okay? Page 1 of 5. Any questions? Examples of polysaccharides. I think glycogen and starch and cellulose are done. Glycogen, storage form of carbohydrates in animals. Starch, storage form of carbohydrates in plants. Cellulose, I think we're done. Okay, don't forget. Cellulose and glycogen and starch, those three, the polysaccharides, they are insoluble in water. The simple sugars like glucose, it is soluble in water and they taste sweet. Anyone has any questions about carbohydrates? We finished carbohydrates and we talked about the food test. We only have fats and proteins and we'll be done. Any questions about carbohydrates?
No.
Okay, very good. Now let's talk about fats. Also, fats are really easy. Now, fats in your body is a very big molecule, actually. Fat is a very big molecule, and fat has another name. It's called fat or lipids. Be familiar with the word lipids. When we eat fats, it will be digested. And we will study the enzymes that digest fat later, okay? It will be digested by enzymes into
Fatty acids and glycerol.
very good, into fatty acids and glycerol. So before you ask me what are fatty acids and glycerol, this is what fat is made up of. These are the monomers or the subunits that make up fats. The fat molecule looks exactly like this. It looks like a jellyfish. This is the fatty acid molecule, and these are the glycerol molecules. You have 3 glycerol molecules. 1, 2, 3. 3 glycerol molecules bonded to the fatty acid. Of course, this has a molecular structure that you don't need to know anything about now because you're still in your O levels. So fats, these are biological molecules that contain carbon, hydrogen, and oxygen only, no nitrogen. And they're broken down in your bodies to give you fatty acid and glycerol. You should not know the molecular structure of fats. You just need to know that it is made up of fatty acid and glycerol, and they look like this. This is the fatty acid and the glycerol. You don't have to need to know even to label it. Just need to know that it looks like that. So in case they give you a picture in paper 2, something that looks like a jellyfish, a 1 molecule and 3 molecules bonded to them, then this is a fatty acid and a glycerol. 1 glycerol molecule and 3 fatty acid molecules bonded to the glycerol molecule. Any questions about fats? Fats made up of 1 glycerol molecule bonded to 3 fatty acid molecules. You don't need to know what is the glycerol. You don't need to know what is the fatty acid. You don't need to know the molecular structure. What you need to know is that fats are made up of carbon, hydrogen, and oxygen. These are the elements found in fats only. This is number 1. Number 2, when they're broken down in your body, they are broken down into fatty acid and glycerol. You don't even need to know the numbers. Any questions? Any question about fats?
No.
Oh,
Fats. Why are they important in your body? Why are fats important?
For making cell membranes.
Yes, we will study more about biological molecules when we study digestion, but it's important to know that fats are important because they are providing us with a storage form of energy. Fats store energy. So we're talking about fats. Fats, they are important in the making of cell membranes. Fats are important in the making of hormones. Okay, fats are very important because they provide us with insulation. What is insulation? They make us warm. Okay, this is your bone. And all your bones and all your organs, by the way, are surrounded by a layer of fat. And this fat keeps you warm. And yes, people that have more fat in their bodies, they are usually warmer than skinny people that don't have fat. So this is the importance of fats. Okay. Who will remind me of the fat test? What is it called?
Emulsion.
Emulsion.
The emulsion test. Good. If I have here a solution unknown, I don't know what it contains, and I want to test it for the presence of fats, what do I add first?
Ethanol.
Ethanol. Another name for ethanol is what? Alcohol. Very good. What's the color of alcohol or ethanol?
Clear.
Yes, very good. It's transparent. And then after I add the ethanol, and I always start with the ethanol, by the way, because ethanol or alcohol dissolves the fat. Okay, I want to break down the fat molecules. And then I add what after I add the ethanol? I add the ethanol and shake. And then I add water. Very good. And then I shake. Now, what will be my result, or what color will I see if fats are present?
Cloudy emulsion.
Very good. It will become milky or whitish or cloudy emulsion. You're all correct. Okay, and we'll watch the video together again. So, any questions about fats? Fats, they contain carbon.
What's the negative result?
Excuse me?
Negative result of fats.
Negative result, it will not form a white emulsion. It will not form a white emulsion. You will not see the whitish suspension. Okay?
What are the triglycerides?
Triglycerides, this is the name that describes the molecular structure of the fat. I've never seen, honestly, a question asking you about triglycerides, so that's why I ignored it. But if you want to know, fine. Fats are made up, my dear, of one glycerol molecule and 3 fatty acid molecules. This structure is called a triglyceride because we have fatty acids. We have 3 fatty acid molecules bonded to one glycerol molecule. That's why they described it as a triglyceride. Okay?
Yeah.
You're welcome. You're very welcome. Okay, proteins. Proteins also, they contain carbon, hydrogen, and oxygen. In addition to that, proteins always contain nitrogen. This is the molecular structure of protein, and I am telling you, don't memorize it. But you need to be familiar with the presence of nitrogen. If they give you several molecular structures and they tell you where is the protein, just go look for the nitrogen. If the nitrogen is there, then this is protein. Good? Proteins, when you eat protein in meat and
Can you repeat this section, please?
of course, proteins, this is the molecular structure of protein. Don't memorize it. Like, don't get a paper and start drawing it 100 times to memorize it. You will do this in AS biology, not now. But how do I know that this is a protein? See, this is carbon, this is oxygen, this is hydrogen, and this is nitrogen.
Elements اللي جواها.
Yes. As long as you see the nitrogen in the molecular structure, then this is a protein. Fine? Before
I
you
have a question.
Yeah, 1 minute. Before you ask me what is this is the R group that makes every amino acid different. And again, this is too advanced. You should not know anything about it. Yes, my dear.
Don't proteins contain sulfur?
Some of them, not all of them, but they all contain nitrogen. That's why sulfur is not mentioned. But nitrogen is mentioned because all of them contain nitrogen.
Okay, thank you.
You're welcome. Okay. Now, proteins are very important for growth and hormones, etc., 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 etc. Proteins, when they are broken down in your body, they give you what? Proteins are broken down to give you amino acids. Okay. To give you yes, what happens here? To give you amino acids. We have 20 different amino acids. 20 different amino acids. How are they different? The only difference in the amino acids is this part of the amino acid. Let me again break it down to you. I've got a whiteboard. So what are we talking about? We are talking about proteins. Proteins, when I eat the protein, the body will digest it and break it down into smaller what? Yes, amino amino acid. What happened to my pen? Okay, now my amino acids, I have 20 types of my amino acids. 20 different types of amino acids. Of course, you will not know the 20 types, but the amino acid, this is the structure of all the amino acids. It is made up of a central carbon atom bonded to a functional group. I don't know if you took functional groups in chemistry. A carboxyl group, COOH.
We took it last year. I think.
You took it, very good. Okay, and then an amine group here. My pen is not working. An amine group. This is the group that contains the nitrogen and a hydrogen and a variable R group. Now let me explain. This part is found in all amino acids. So this is a fixed part. This R group is known as the variable R group. Sometimes my pen will not work, okay? This is called the variable group. Why? Because this is the part of the amino acid that is different from the other amino acids, and this is what makes amino acids different, okay? So this group is the group that contains the nitrogen. It's called the amine group, and this is the group that is removed by your kidneys. We'll study it in another lesson. Let's go back to proteins. Proteins. They are made up of amino acids. We have 20 different amino acids. They all contain the same basic structure, but the R group is different for each amino acid. When amino acids are joined together, they form a protein. The amino acids can be arranged in any order, resulting in different proteins, hundreds of proteins. Even a small difference in the order of the amino acid results in a different protein being formed. The DNA determines the order of the amino acids in proteins. Now let me explain this more. Remember when we studied the DNA and we said that the DNA has different base sequences, and these base sequences code for amino acids?
Nitrogen bases?
Yes. Now again, I'll explain the DNA, okay? This is actually the supplement part of the lesson. The DNA, or the chromosomes, is made up of, I think I explained this in class, but I'll explain it again. It is made up of a sugar molecule bonded to a phosphate molecule and a nitrogen base. Okay? This sugar and phosphate molecule is found in all our DNA. What is variable? The nitrogen base, the order of the nitrogen bases. Now, these nitrogen bases, when they are in a particular order, they will be converted into amino acids. This is an amino acid, a small protein molecule. Many amino acids bonded together, what do they make?
Protein.
Proteins.
Protein.
Fine. Now, I think you remember that we said that we will compare DNA of the different organisms. What exactly do we compare in the DNA?
Basic.
The base sequence. Very good.
Arrangement of them.
We also said that I can compare the proteins in my body and the proteins in your body. What will make the proteins in my body different from the proteins in your body? The order of the
arrangement?
The arrangement of the what? Proteins are made up of what?
Ribosome.
Amino acids.
Amino acids. Very good. So again, I'll try to make it as easy as possible. The order of the bases determine the amino acid, and the order of the amino acids decide which protein will be produced. Let's give easy names so we understand. Let's say we have 3 amino acids giving us a particular protein. These amino acids are in this order: 1, 2, 3. Amino acid 1, you don't have to know its name. Next to it, amino acid 2. Next to it, amino acid 3. This will give me a particular protein. If I change the order of the amino acids, a completely different protein will be made. For example, if I put amino acid 1, and then amino acid 3, and then amino acid 2, I will create a different protein. Okay? So what is this part of the lesson trying to tell you? That the proteins are made up of amino acids. And the order of the amino acids decides which protein will be made. If the order changes, the protein will change. Anything hard about what I said?
Doctor,
Any questions?
what is DNA made of again?
DNA is made up of nucleotides. Can you leave DNA now? Because we will take DNA tomorrow in class. I'm talking about proteins. Did you understand what are proteins made up of? Who was asking?
Yes.
Okay, proteins are made up of what, my dear? Smaller units. These smaller units are called what?
Amino.
Amino acids. Now, when the order changes, what will change?
Protein.
Proteins. Any questions? Anyone has any questions concerning proteins?
Yes, the
I have
amino
a question.
What is your question?
What is the R group?
I'll tell you what is the R group. Let me just open a PowerPoint because I need something to scribble on.
Miss,
Yes.
the amino acids, the base sequence of what depends our
The base sequence of the DNA. Let's go back to the very beginning. We have a cell, and this cell has a nucleus, and we have chromosomes. Okay? I'm going to get out the chromosome, and I will look at the DNA inside the chromosome. The chromosome is made up of DNA. Whoever was asking about the DNA, pay attention. The DNA molecule, my dear, is made up of this structure. It's made up of a sugar and a phosphate and a nitrogen base. And a sugar and a phosphate and a nitrogen base. So you can see that the nitrogen bases are arranged in a certain order. Yes? I have nitrogen base 1, nitrogen base 2, nitrogen base 3. Don't you see that? Who was asking? Who was asking?
the amino acids.
Very good. Now these bases, they make amino acids. When the order of the bases change, they will change the amino acid being produced. So the order of the bases determines which amino acid is being produced. Clear?
Clear.
Good. Now, talking about the R group. The amino acid, how does it look like? The amino acid looks like this. It is made up of one central carbon atom bonded to a carboxyl group, bonded to an amine group. I don't want you to memorize it, okay? I'm just answering your question. Bonded to a hydrogen here, okay? And bonded to an R group. What is this R group? Because someone was asking. The R group is the variable group. What do I mean the variable group? My pen is not working. I mean the group that is different, that makes every amino acid unique, that makes amino acids different from each other. This part of the amino acid is constant. All amino acids have this part. What makes amino acids different? The variable R group. Whoever was asking, is this clear now? Is the R group clear now for everyone?
Yes.
Okay. Now, going back, I don't know why is this open? So, we're done with proteins. What is the test for proteins? Who will remind me?
Benedict solution.
Not the Benedict. The Benedict is for the reducing sugar. That burette, correct. Now, where is it? Here. This is the test for protein. We will add drops of burette to the food sample. Fine. A positive test will show a color change from what to what?
Blue to purple.
Blue to purple. So this is my food sample, my unknown. I'm going to add the burette solution. And before you ask me what is it, this is a ready-made solution in the lab. I'm going to add my burette that is blue. If protein is present, it will change from blue to purple. If protein is not present, it will not change from blue to purple. Clear? Now, any questions about proteins? Any questions about proteins? Are proteins clear?
Yes.
Yes, they're clear.
Good. Again, proteins are made up from amino acids. The amino acid has a particular molecular structure. Carbon bonded to a carboxyl group, bonded to an amine group that contains the nitrogen, bonded to a hydrogen group. This part of the amino acid is fixed. It is found in all amino acids. The variable part is the R group. This is what makes amino acids different. And we have 20 different amino acids. Amino acids, when they join together, they make polypeptides. Polypeptides means proteins. And they have a test, the burette test. I'm going to add the burette, drops of burette. I'll shake, it's blue. If protein is present, it will change to purple. This is all what you need to know about proteins. Any questions? Okay, now what is this picture? This picture, this small picture here, is a apparatus that is found in the lab, and you have something similar to it in your kitchen. It is used to crush the food, because if we are doing any food test on a solid food sample, I first have to crush it. Okay, so that I can carry out my food test. The only food test that does not need crushing is the iodine test. Look here. I can add drops of iodine on my potato directly. I don't need to crush my potato. And it will give me a positive result from yellow-brown to blue-black. But if I have an ethanol test or a reducing sugar test and the sample of food is a solid, I first have to crush it. And this can be a question in paper 6. What do you do with a food sample if it is a solid? Can I immediately apply the Benedict, or I have to crush it first? I have to actually crush it with a little bit of water to get out the soluble products and filter it and use the filtrate. It's enough to say that I will crush it and I will crush it with a little bit of water so that I turn it into a solution. And then I will start my food test the normal way. Clear?
Yes.
Okay, let me see what else is in this lesson. This is actually a very easy lesson. Let us read together word by word. If the food sample is a solid, it must be crushed with some water to extract the soluble products. Then this food is crushed with a mortar and a pestle. Before you ask me what's the mortar and the pestle, this is the mortar and the pestle. This one. This is the mortar and the pestle. Here. I have a picture here for you so that you know what is it. The mixture is filtered and the test is done on the filtrate. I'll filter it. I will use the solution that comes out, which is called the filtrate, and I'm going to do the test on this solution. Burette is a strong alkali, so you must be wearing gloves during the experiment as a safety precaution. They will ask you, what is the safety precaution? I have to wear gloves. I cannot touch any of these chemicals in the lab, even if they're safe. I have to always protect myself by wearing gloves. If it touches your skin, wash it off with water immediately. Okay, these are instructions that you should know about in the lab. And also, the water bath in itself is a safety precaution. Okay, because I want to avoid the direct flame. I want to avoid the fire. I don't want to use the fire in the lab because it's very dangerous. That is why I will heat my Benedict in a hot water bath. Using a water bath for heating is in itself a safety precaution. Clear? Anyone has any questions? Okay, now pay attention to this. In one of the questions in paper 6, they told you that we have seeds. And we want to test those plant seeds for ethanol. So I want to test those seeds for fats, So we will do the ethanol test. Can I just add the ethanol directly to those solid seeds?
No, we need to crush it
I'll
first.
crush first.
Very good. I have to crush it. But this time I will crush it with water or I will crush it with ethanol.
Ethanol.
Very
Ethanol.
I will have to crush it with ethanol because I want the fat to come out, and the fat and the ethanol or the fat dissolves in the ethanol. So if the solid sample or the seed is tested for the presence of fats, It must be crushed with ethanol, and ethanol is marked in yellow, to extract the fat-soluble products first and then filtered. And then whatever comes out, the filtrate is then tested for fats the normal way. I will add ethanol again, I'll shake, I'll add water, I'll shake, and it will become, or it should become, cloud. Any questions?
No.
Good. Now the test for the vitamin C. Again, we said it in the lab, but we'll say it again. What's the test for vitamin C called? What's the reagent that we will use?
DCP.
DCPIP. Here I will start with the DCPIP. I will put my DCPIP in my test tube. See? I will put it in my test tube. The starting color is what?
Blue.
Blue. And then slowly I will add drops of the vitamin C. I'll add a drop and shake, and drop and shake, and drop and shake. And if vitamin C is present, it will gradually become or turn into colorless. Okay? If I add one drop and immediately it changes from blue to colorless, then this shows me that the vitamin C was concentrated or dilute?
Concentrated.
Concentrated. Very strong. It has lots of vitamin C. Just one drop changed the DCPIP from blue to colorless. And you don't have to know what the DCPIP stands for, because some of you will ask. Okay? When describing the food test in exam questions, this is very important. Make sure you give the starting color and the final color. No crushing is involved in liquid food samples. If you have a liquid food sample, you don't say, I will crush it. This will be very silly. What will you crush? It's a liquid. Or in iodine or starch test. Only if it's a solid sample like a seed, then you have to crush that. Okay? For the iodine or the starch test, you don't need to crush anything. Iodine can be directly placed on the solid food sample, such as potatoes, and it will change color. Any questions?
No.
Okay. Where are you going? Wait. We will just revise to make sure that we're done with this test. With this lesson. It's a very easy lesson, and it's very short. We have biological molecules. We here discussed, or we talked about carbohydrates and the proteins and the fats. Now you remind me, what are the two types of carbohydrates?
Simple and complex.
Very good. Simple carbohydrates, give me two subdivisions. If it's made up of one glucose unit, it's called a mono or a disaccharide.
Mono.
Disaccharide. Okay, let's write notes together. Here, carbs. I hope my pen works. It's not working. Okay, we have the simple, no, I don't have a pen. The simple and the monosaccharide. Okay, give me an example of a disaccharide.
Sucrose and maltose.
Sucrose مع maltose.
Excellent. What is the test for the reducing sugar?
Benedict.
Benedict.
Benedict. Okay, we have to heat it here or we don't?
No.
We have
No.
We have.
Yes.
Yes. Yes, Benedict test is the only test that needs heating. Don't forget. Okay. Now, what is the test for starch?
Iodine.
Give me three
Protein.
examples of polysaccharides.
Cellulose.
starch,
Cellulose, glycogen.
and glycogen.
Good. Which one is the starch form of carbohydrates in animals?
Glycogen.
Glycogen.
And which one is the starch form of carbohydrates in plants?
Starch.
Good. Cell wall is made up of what?
Cellulose.
Cellulose. Okay, you're more than amazing. What are the elements found in carbs?
Carbon.
Oxygen.
Nitrogen and oxygen.
Excellent. Do we have any nitrogen?
Yes and no.
No.
Okay. Fats. Fats are broken down in our bodies into what?
Energy.
Fats and glycerol.
Okay, I did not hear any of you, but you're all correct also. Fatty acid and glycerol. Okay, if you want to know the molecular structure, it's one glycerol and three fatty acids. That's why they call it triglycerides, but you don't need to know these details. Fats. How do we test for fats?
Ethanol.
Test. Why are fats important in our body?
Margin test.
Yes, you are correct. Why are fats important in our bodies? Fats are used for what?
Store energy.
Making
Insulation. Cell membrane.
Yes, you are all correct also. For cell membranes, for storing energy, to making hormones, to insulate our organs, to keep us warm, etc., etc. Good. Proteins. What are the elements found in proteins?
Hydrogen, nitrogen.
Hydrogen, oxygen, and nitrogen.
Okay, perfect. They are broken down to give us
Amino acid.
Amino acids. Perfect. We discussed a lot of details, by the way. We dived into advanced information that you don't have to know, but I was answering your questions. But proteins are broken down to give us
Amino acids.
amino acids. How are amino acids different? What is the difference between one amino acid and the other?
The R group.
The R group, yes.
The R group.
Very good. Perfect. Okay, we're actually done. This is the core part of the lesson, but the core part is very important. Yes, we forgot. What is the test for vitamin C?
DCP.
DCP. What is the starting and the final color?
Blue and colorless.
Okay, don't forget, when you describe the test for vitamin C, start by adding the DCPIP in the test tube. Not the vitamin C, the DCPIP in the test tube, and I keep on adding drops of vitamin C and shaking. Drops of vitamin C and shaking, and it will slowly change from blue to colors. Okay.
Drops of food
Yes,
or
my dear.
vitamin C?
Huh?
Drops of food or vitamin C?
The unknown food sample. We want to know if it contains vitamin C or not. If it does, then the color will change from blue to colors. If it does change from blue to colors, then yes, it does contain vitamin C.
Okay, thank you.
You're welcome. Anyone has any questions?
No.
Okay, very good. See you tomorrow.
Okay. Goodbye.
Goodbye.
Bye.